The invitation to give this webinar at LabRoots. I'm going to talk about our recent and still pretty new efforts to establish a new model organism for the neuroscience of vocal communication and auditory processing of social cues. And as you can see from the title, the organism is going to be the Mongolian Gerbil. And for those who don't know, this is actually already a well-established rodent species for studying hearing loss and other sorts of auditory processing. And it's also been studied by field biologists quite a lot. So I'll go into some of that background in a few slides. The real contribution of our new work is to try to get the best of both worlds by establishing a large and semi-naturalistic environment in the lab where we can study the gerbil's natural behaviors, but in an environment where we can make more complete measurements, including eventually neural recordings in auditory cortex. And as I mentioned, this is still recent and developing work, so I'm excited to give you all this kind of early look into what we've been working on. So backing up a little bit, I think most neuroscientists are interested fundamentally in understanding how the brain gives rise to adaptive animal behaviors. And before getting into the specifics, I think it's worth pausing and appreciating how challenging that problem actually is. And in fact, behavior is so complicated and multifaceted and maybe even ill-defined in certain cases that neuroscientists have traditionally done everything in their power to place constraints on animal behavior in order to study it more precisely. So these take the form of physical constraints, such as head fixing. This can help us get more stable neural recordings, but it also prevents the animal from making uninstructed or complex movements. Also, neuroscientists, traditionally at least, have been very focused on training animals to perform precise and repeatable behaviors, such as reaching to grasp an object or moving a joystick with a small number of degrees of freedom. So these are very powerful approaches because they give us a lot of precise control over what the animal is doing, and that helps us draw connections with how the brain is underlying those specific behaviors. So this is going to continue to be an important approach, but ultimately understanding the brain, I think all of us would agree, is going to require us to move beyond those settings to understand neural activity under more complicated and realistic natural settings. And this is obviously critical if we want to develop translational clinical therapies or neuroprosthetics that work in the real world. And I would say in the last decade or so, the idea of studying natural, untrained, and unrestrained animal behaviors has become more and more popular in systems neuroscience. So the NIH Brain Initiative has identified this as one of its central goals. And there has been several influential perspective pieces making this basic point, two of which I'm citing on this slide. So overall, I think the basic idea is quite intuitive and sensible because we've finally developed the experimental hardware that's necessary to record neural activity in animals that are freely moving in a complicated environment. And we're also developing the computational software and analysis tools necessary to do things like track animal body position with high-speed video and process all that data into something that we can interpret. So those are the trends within the broader field that we're kind of riding on top of. What I'm going to tell you about today is our efforts to study natural animal behavior that has a strong auditory or acoustic component to it. And my personal interest in this as somebody who develops computational tools and data analysis pipelines is driven by our relative lack of computational tools in the auditory setting. So in particular, there's a lot of groups working on, for example, tracking animal body poses using video data or multi-camera arrays. But I think there's still a lot of open problems and relatively fewer people potentially working on solving analogous problems for complex audio measurements in environments where there are multiple animals making noises simultaneously overlapping. So on this slide, I'm just juxtaposing a picture of a hearing aid and a crowded room of people talking to make the point that complex social environments are important for auditory neuroscientists to study if we want to develop better clinical technologies down the road. So hearing aids work pretty well, but generally speaking, they're optimized for hearing in much simpler artificial and controlled settings, the more traditional experimental setting that I showed on the first slide. And we're trying to take steps towards understanding or adapting these technologies to more complicated systems. So in particular, we're going to focus on a particular kind of auditory information transfer called vocal communication. So this is widespread across the animal kingdom, but comes at very different levels of sophistication across species. So on this slide, I'm showing you 3 example model organisms that are known to have a pretty sophisticated vocal repertoire, and those vocalizations are really important for their survival and reproduction. So male zebra finches, for example, learn a sophisticated courtship song that is necessary for them to reproduce. And because that's under such tight evolutionary pressure, they vocalize a lot, and they do it in a stereotyped manner that helps us study it. So overall, there's a lot of model organisms that would be interesting for us to study. And I think it's important for neuroscientists to think about this diversity across different animal species and how they communicate vocally. And it's useful for us to study animals that naturally have a complex vocal repertoire and naturally want to vocalize a lot. It's not a behavior that you really can train easily in animals. You need to pick your model organism in a smart way. So that brings us to the Mongolian gerbil, which our team thinks is going to be an ideal model organism to study vocal communication. And the reasons for this is that they have a rich social structure that is really built on top of vocal communication. So fieldwork has established that gerbils live in 10 to 20 member families within the wild. They're pair bonders, and they are cooperatively parent new pups. So it's not just the mom and dad that help rear the pups, but actually all the older siblings that stay within the family help to raise the young. So they're a highly cooperative species, and there's a lot of evidence that they can distinguish family members from non-family members. So there's low rates of breeding within the family under natural settings. And also they live in these broader neighborhoods of many families. And if gerbils from a different family enter the territory of a different family, they're recognized and usually kind of forced out. So a lot of interesting social behaviors within this cooperative mammalian species, and a lot of them are driven by vocalizations. So later I'm going to show you some results that we have within our lab, and we show that the vocal repertoire of this animal species is pretty sophisticated. You can detect or you can distinguish which family is vocalizing based on the statistics of the vocal calls, for example. Okay. So as I mentioned, this is a species that's been of interest to field biologists for some time. So this is a picture from a relatively recent review showing a burrow system of an example family. This is actually a relatively small burrow system. If you look throughout the literature, you can see that these burrows can actually be quite extended. And it makes sense if they're holding up to 20 family members or so. So there's a nice literature that we're building on top of in terms of the natural ethology of this species. And also there's been a strong interest in gerbils within the specific field of auditory neuroscience, or especially neuroscientists studying hearing loss and wanting to draw connections to humans, because gerbils hear frequencies that are much more similar to the human hearing range compared to, for example, mice or rats or other more common laboratory rodents. So this is another reason why we think gerbils would be an interesting species to study in the context of natural social behavior, because there's been a lot of kind of low-level sensory neuroscience work within auditory cortex. And more recently, within the last 10 years, a lot of the modern tools that you would want to see in a organism that you're going to use for neuroscience research have come online. So in particular, there's now a standardized anatomical brain atlas that we can use to figure out where to implant electrode probes. The genome of this species has been sequenced, which opens up, the possibility of many future experiments down the line. And as I already mentioned, this is a well-established model for studying the neurophysiology of hearing loss. So we're able to build on top of all of these existing bodies of work to study them in a natural context. So with that, I want to briefly introduce our team before telling you some of the early steps we've taken towards establishing this as a model organism in the lab. This is mostly work that's been led by a very talented grad student, Ralph Peterson, and he's been helped by a number of people, most primarily Aramis, Amand, and Nigel. And there are, in addition to myself, who's the PI leading the computational component of this project, I have two co-PIs on this project, David Schneider and Dan Sains, who focus on the experiments and are experts in auditory neuroscience. And there's many, many other people that have helped get us off this line. I'm just showing six people here. But this list is rapidly growing because it turns out that you need a lot of cooperation among humans to build up a large naturalistic environment to study cooperative animal behaviors. Okay, so I'm just going to give you a couple tidbits from our first observational study that is now available as a preprint in the journal eLife. So what Ralph did here was he developed a somewhat semi-naturalistic environment, much larger than a typical environment that you would house gerbils in, but still much, much smaller than what you would expect to see these gerbils living in a real environment. So this is what we're calling kind of version 1 of our semi-natural environment, and currently we're working on version 2, which is several times larger than this and will include things like a burrow system. But already, even just within this environment, we found some interesting things. So what Ralph did was record through a microphone array and overhead camera, the 24/7 behavior of a family of gerbils. So mom, dad, and 4 pups. And he did this across 3 families, and for each family recorded for a month. And we picked a developmental time window that we thought would be particularly interesting, which is when the pups are still in the critical period for auditory cortical plasticity and up until the point where they're about to be weaned. So we reasoned that there would be interesting vocal dynamics that change over the course of several weeks as the pups develop through this phase. Indeed, when you put the animals in this setting, they vocalize like crazy, and they naturally perform a lot of behaviors that you wouldn't see if you house them in isolation, for example. So just to give you some rough sense of the scale of this data, each one of the gerbil families produced roughly 10,000 calls per day. So that was a total of over 500,000 calls in this data set that we were able to detect and extract, and 13 terabytes of raw data. So Ralph did a lot of work to comb through all of that and summarize what we found in this first study, and I'm going to give you just a few of the highlights without going too deep into the details. So the main questions that we want to answer with this approach, and especially the version 2 environment that we're working on now, I would say the three main questions are, first, what is being said? So what are the vocalizations that the gerbils naturally produce? Second, who is saying what? So once we have a sense of what the natural vocalizations are, we want to know how they're used and whether they're differentially used by mom, dad, older pups, younger pups, etc., and under what circumstances. So in order to do that, we need to attribute each vocalization to one of the individuals. And then finally, we want to bring this back to neural recordings and auditory cortex. And I'd say that we have a reasonable first approximation to the first question, which again is available as a preprint. And the second two questions are things that are still in process and we have preliminary data for. So for today, I'm going to focus on just the first two because these are maybe the furthest along. And again, give you a kind of eye towards the future of where we hope this project is going to go. Okay, so this is the preprint on eLife that Ralph put together for our first observational study. And just to jump to the conclusions of our work without going into the details, I'll tell you that what we found is that more or less, if you had to ballpark it, there are 15 to 20 major vocal call types that are expressed in the environment by the family. And within each one of those vocal calls, there are kind of variations. So if you include those sub-variations of the main vocal calls, we ended up using something along the lines of like 70 microclusters of different vocal calls. And we used that to quantify how the vocalization statistics were changing across families and across time. So we also see that the vocal calls occur in bouts, and I'll show you an example of this in a few slides, where it's not just an animal giving a single vocal call, but really they'll produce a vocal call, and then oftentimes they'll repeat that vocal call or a similar vocal call multiple times. And as I mentioned already, these vocal calls are dynamic and changing. So they change. We find that they change over the course of development as the pups are raised from, young to their day of weaning. And they also change. There's a very strong circadian effect. So the kinds of vocalizations you see at the beginning of the day are very different than the vocalizations you see at the end of the day. And we also can reliably detect differences in the vocalizations across families. And, we might speculate that these would be dialects that the families naturally develop. And those differences in dialect might be useful for preventing incest or recognizing strangers, as I was alluding to before. So just to give you a couple of the figures from this paper, here's a figure showing a histogram of the vocal call statistics across two families, family 1 and family 2. So there's 70 different clusters here. Each cluster represents a kind of very specific variant of vocalization. And you see that some of these vocalizations are used much more prominently in family 2 than family 1, and vice versa. And we can statistically quantify this and show that this is much more diversity than you would expect by chance. I also mentioned the idea of vocal bouts. So here are 3 example vocal bouts that we recorded. I'm showing you the spectrograms over time, and you can see that there are vocalizations in each of them that are being produced. And what tends to happen is that a similar vocal call is repeated multiple times. So you can see that in all 3 of these examples here, the spectral statistics of the vocalization look very different across the 3 bouts, but from vocalization to vocalization, they look much more similar. And I'm glossing over the details of our analysis for how we actually pull out these clusters. You can read about those details in our paper. But just to give you a sense of how the analysis works or some sanity checks that you can run, I'm showing you a low-dimensional projection of the data on the right, where each point in this low-dimensional space represents one of a vocalization in the data set. And I'm coloring the points in this low-dimensional space by which bout they came from. So you can see all the purple dots, which came from the first bout at the top, clustered together in this auditory feature space, and likewise for the green dots and the yellow dots, which are the other two bouts. So this is just showing that we have an unsupervised method that helps us cluster the vocalizations, and you see temporal regularity within these clusters, which is a good sanity check. Okay, so that's a rough overview of what is being said. There's a lot more that I could go into for that story, but for the last 5 minutes or so, I want to give you another angle that is a more recent part of this project that has been in development. And this has been the question of not only do we want to know what is being said, but we want to know at each point in time which gerbil is saying that, so we can begin to understand what is the behavioral purpose or the effect that is propagated by that vocalization. So this problem of who is saying what has been studied within the field of acoustic engineering for a while. This is sometimes called a speaker identification problem. People have been developing algorithms for this in human settings. You can imagine a conference room of people talking to each other, and you want a transcription of what was said and who was saying it within the conference room. So we're effectively trying to take ideas from acoustic engineering and audio machine learning, modern methods from machine learning, to solve this problem in our laboratory setting, which has a couple of distinct features. So this, what I'm showing you here, is just what we hope to achieve by developing these methods. So again, we would have a group of animals within a semi-naturalistic environment. We would get a recording of what vocalizations were being produced, and for each vocalization, we would be able to tag it to one of the animals. And effectively this goal is trying to fuse together our video camera information with the auditory data. So we have two streams of data, and we would like to make sure that they're self-consistent and cross-referenced to each other. So the reason that we're able to pick up who is saying what, or the reason we have some hope of solving this, is that we have multiple microphones throughout the environment, and you can pick up differences across those microphones based on where the animal that produced the sound came from. And there's two main kinds of differences. The most classical kind of difference that the acoustic engineering field has used is phase differences or time of arrival differences. So because the speed of sound is slow enough, you can pick up those differences in arrival times across microphones, and then have an inference about where the sound originated from. And the second kind of difference would be a level difference across the microphones. So if the animal is close to one of the microphones versus another, then you would imagine the volume that there should be a volume difference across the microphones. And interestingly, what the kind of classical methods in acoustic engineering and the methods that neuroscientists have first started using for this problem really don't pay attention to volume differences, and they instead pay attention to phase differences or time of arrival differences. And keep that in mind, because in a few slides I'll show you that it might be more important for us to think more carefully about the volume differences. So we've tried several approaches at this point, several machine learning methods, and one approach that seems to work pretty well is to use deep neural networks to solve this problem. And indeed, if you look at the machine learning field, this seems to be where people are going. So the basic idea is to detect vocalization events and then take snippets of the audio from all four microphones and put them through a convolutional neural network and have the network predict the spatial location where the sound came from. And we collect training data sets where we know the ground truth. This can be collected either by having a robot move through the environment with a speaker, so you get sounds produced from many different areas of the environment, or we have a way of actually getting isolated gerbils to produce vocalizations by playing conspecific vocal calls over the speaker system. So we collect a large training data set with ground truth labels. We train this deep neural network, and then we have it output predictions on held out data. And one of the really important things that we immediately realized is that perhaps it's even more important to have a network that gives you a good estimate of its confidence or uncertainty than being a very accurate network. So this is the idea of calibration. So when we train these networks, we train them not just to output a point estimate, but to actually output a probability distribution over the 2D space. And what we found is that not only is it helpful to train individual deep networks to do this, but you can train several deep networks in parallel called an ensemble of networks and ensemble their output predictions. So at the end of the day, what we get out of these models is a 95% confidence set effectively. And what we can do, this is what's shown on the right part of the slide, is make sure that this confidence set is accurately calibrated so that 95% of the time, the true vocalization is actually within that confidence set, and 5% of the time, it's going to be outside the confidence set. So this idea of having a well-calibrated model is something that statisticians and machine learning practitioners care deeply about. And it's something that I think is not yet used rigorously in this particular setting in neuroscience. So this is one thing, one innovation that I think is very important and important for us to get right. So there's details about how you make sure your model is calibrated and how you can encourage it to not be overconfident or underconfident that I'm going to gloss over, but this is an important part of our approach. And jumping to the punchline a little bit, what we find is that this works pretty well. However, the deep networks, in order to learn this, need a lot of training data. So on this plot, I'm showing you the amount of training data you get versus the loss or error of the model. So that error is going down. This is a log-log plot, so that's kind of going down like a power law. So you need to collect thousands or tens of thousands of vocal events to get really good performance. So this is a challenge. So as I mentioned briefly before, our approach to overcoming this challenge comes from collecting large ground truth data sets where we move a robot around this environment and have it give vocal calls. And also another approach that we've been piloting more recently is to create a lot of simulated data using a highly accurate computer simulation of audio environments. And with all of this, we can, at the end of the day, within our current version 1 environment, we can also often localize sound sources within 1 centimeter of their true source. So here I'm showing in blue the cumulative error distribution of our deep neural network, and I'm comparing this to Muse, which is a method that uses phase differences across the microphones and is based on kind of more classical acoustic engineering principles. So again, I kind of hinted at this idea that these classical approaches to solving the speaker identification or sound source localization process, solving those problems relies on phase differences in the microphone signals. This is known as beamforming in the engineering literature. And what's interesting to us is that the deep neural networks in our environment seem to ignore the phase differences, and instead they're paying much more attention to the volume level differences. And we showed this in a number of ways. I'm not going to show you all the results here, but here I'm showing you the simulated data set using this Pyroom acoustics package that I mentioned a few slides ago. As you increase the amount of reverberation within the environment, what we find is that the deep networks are paying much more attention to volume differences versus phase differences. So the way to read this plot is the x-axis is showing you the wall absorbance. So as you move to the right, there is less and less reverberation within the environment. And then the problem is easier. So this green line is the beamforming kind of classical approach. It actually works pretty well. But in our real environment, we think there's a lot of reverberation. So we're more towards the left of this plot. And there, this green line, which is the classic model, has a much higher error to the deep net models, which are able to use, basically ignore the phase information because it's less reliable and use other cues to solve the task. Okay, and finally, I would just say that, again, we're in early days of solving this problem. We think that we have a good pilot solution to it, but we need, in order to make it very robust and reproducible across labs, we need to still do a lot of work. So we've been working on developing, collecting and curating a large number of benchmark data sets for this sound source localization problem across not just the environment that we're looking at, but many different environments, different microphone configurations, different animal species, et cetera. So some of the data sets that we've collected here are shown on this slide, and they're already available online if you go to our GitHub. And this also includes all the code that we used to train the models. So we're hoping to share all of this to kind of jumpstart a community effort towards solving and tackling this really challenging computational problem. And with that, I just want to thank once again everybody who's worked with us and the NIH for funding this. And I think that's it.